Okay, so I'm Jim Collins. I want to thank George, Nancy, Andrew, who just left, and Jeff for inviting me to spend 420 seconds telling you a little bit about biocontainment. And specifically, I just want to highlight, it's too bad Farron Isaac's ill and can't speak here. He was going to speak before me and talk about recoding genomes and using auxotrophs as a safety mechanism. I'm going to talk from now bottom up and just very quickly mention some work we're doing and to challenge folks to think about how you can use small synthetic circuits as safety mechanisms. We, for the beginning of the field, have been playing around with how you can actually model, build, and construct different circuits and use them to rewire organisms. Going back about 6-7 years ago, we got excited of doing very simple ones, and I'll show you initially ones we've done in microbes and close with one in humans, where we did a very simple two-input AND logic gate using tight RNA riboregulators we had designed, that if you then had two different molecules to control this, when you introduced those, you would express lytic proteins that could rapidly and readily wipe out a microbe. So in this case, you could have a system where you could deliver a molecule, wipe out something that maybe was in an environment that you no longer wanted it in that environment. As we begin thinking about biocontainment, there's been a lot of interest in the field now the last few years. Could you program organisms so that when they left the environment on their own, they'd be programmed to self-destruct? And I'll share two newer switches. One is a so-called dead man switch. It's a fairly complicated looking circuit, but it's basically an imbalance toggle switch. So toggle switch typically presented as a bistable system where you have co-repressive genes, bistable in that you can exist in one of two states, either gene 1 is on gene 2 is off or gene 2 is on gene 1 is off. In this case, it's an imbalance toggle, which is much easier to make than a bistable one where one side's stronger than the other. And you engineer that one so that you keep it in the unstable state with a molecule that's present in the environment that you're trying to keep your system in. If it leaves that environment, doesn't see that molecule, now the stronger state has a chance to come on. When that comes on, it's going to relieve repression to express a toxin. In this system, we also engineered a protease degradation system that, when flipped, would speed up the switch and also target one or n number of essential proteins. When we had then that combination of a toxic protein and degrade essential, we got beautiful, very quick, and in some cases, sterilization of the culture. Now, what we've done is something more complicated. In the interest of time, I'm not going to be able to walk through it as carefully as I like. But we've also made what we call the passcode switch. So now this is a more complicated scheme, easiest to kind of follow it by looking at the logic diagram on the far right. It's a system that's controlling the expression of a toxin. You've got three different repressor proteins controlling different elements of the switch. The best is the guy at the end. You've got C. You want to make sure C is present in order for your bug or your organism to live. And so you better not have your inducer for C. So C can't be there to have survival. You then have two other repressor proteins that in this case are going to control the expression of the repressor protein that keeps the toxin off. You want to make sure that in this case, basically these guys are not present to shut that guy off and allow the toxin to come on. So you got to make sure that both input A and input B are both present. Okay? Driving this is a thing that I think we as a field need to continue to challenge, is that as much as there's this marvelous synthesis work and more and more folks getting in the field, we still have a very small number of parts to play with. And there's a challenge to come up with better parts, harvest them from organisms and designing them. And part of what drove this was the recognition you could actually design in a mix and match fashion hybrid transcription factors. Anyways, just quickly, what we did was you now actually put some in. And here we're using three different hybrid transcription factors, one that can be induced with IPTG, one that can be induced with GAL, and one with cellular BIOS. It's a three-input system, each with two choices, yes, no. So it's 2 to the 3. You have 8 different possibilities, one of which leads to survival, the other of which are containment for your system. So the survival would be, you better make sure you don't have IPTG, 0. You better make sure you have both of the inducers, GAL and cellular BIOS. Any other of the 7 combos will lead to death. The beauty of this from a security standpoint, IP or espionage, is you can readily change with the passcode. You can expand this quite easily as well. And when we actually implemented this in E. coli, we got brilliant results. So that green in this case means your bug survived. And we found that if you did not have the right passcode, these bugs readily and rapidly killed. An appreciation that is only now just growing in SynBio is that most of the stuff we make is not stable in the way that we think. That is that, yes, it functions nicely when you're doing your experiments for your grad student, your postdoc, but when you look out for a longer time and you begin thinking about, you're going to put this in somebody's body, you're going to put it in the environment, you're going to play with it in a bioreactor or in your lab for a long time, most of this stuff really doesn't work for very long. Oh my gosh. I was wondering how you were keeping time. I was told there was no clock, so I thought I would just be irresponsible. Quick, but irresponsible. Anyway, so I'll go through this fast. We actually did analyses looking out on these circuits in time, and the dead man switch lost stability pretty quickly. It was fine after about a day. Most of the time, you actually had mutations where you had transposons were being inserted, different components. The passcode was a little better. It could go out 2 days, maybe 3 days. And more critically, though, and it's a challenge for George and for guys like Jeff, where if you recoded it, if you actually got rid of the ability of the bug to introduce transposons, if you reduce its ability to mutate, repair those mutations, we could actually significantly stabilize the system. Okay, so it's a thing I think we as a community need to think about, whether from the biocontainment, but also for all these other functions that people are thinking about putting in, will it function as you like? And our field, for the most part, is still in the microbial world. Some great stuff you hear on bacteria. Jeff and Patrick are leading marvelous efforts in yeast, along with Tom Ellis and others. But much of this is also, can you move to higher organisms? One of our fearless leaders, George, and what we did a few years ago is actually produce a really strong kill switch, safety switch for human and broadly mammalian cells. So this was a really nice system where you've got two repressor elements. You've got a repressor protein that keeps something off, and then you actually have an RNAi component that if you get any leakage, it knocks it down. Tara Dean's designed this in our lab, and a great demo of machismo that she did is she actually used it to control the expression of DTA. This was broadly made as a tight gene expression system. Used it to control DTA, so the theory toxin, single molecule which could kill a cell. And she created stable cell lines with this. She was able to grow them up and then selectively turn it on as she'd like, and she could rapidly and readily kill off the cell. So I think it's an interesting tool as we also think about moving beyond cell cultures to living organisms, how I could use this also in a systematic way to make model organisms with different functions. And with that, I'll end with this network of higher organisms whose work I got to share with you. Thanks again for the 7 minutes. Yeah.
Does this sort of system require that all the products be non-autonomous?
To be,
what's the non-autonomy or autonomy requirement of the products for this system to work?
you mean autonomy relative to the rest of the genome?
Export, so these all have to be products that are kept within the cells versus can be exported.
From the cell.
Yeah.
So you certainly could engineer them so that they could be exported from the cell, absolutely, and in fact have it so that they could kill off other guys in the neighborhood. The challenge in some of these, bacteria it's easy, right? You could actually have it so it's lytic, and lytic you'll now have the killing agent can easily go around. Otherwise, we have actually found, and Tim presented some nice stuff on synthetic probiotics earlier, it's not as easy as you think to get a lot of stuff out of some of these organisms. But in many cases, some of the stuff we've done, we've actually produced antimicrobial peptides that tend to like to be on the outside to kill, delivered with phage mids, but they then lyse, and they actually act from the outside then.
Thank you.
Sure.
What about unintended transfer from cell to cell?
Yeah, so unintended transfer, Mark asked about. We haven't seen that yet on this, but you can envision that if you had a protective element, right here you're trading handguns, self-handguns, really, on this. So the cells generally don't like what we have here because it's going to kill them, right? They're suicide modules. Nonetheless, I think it's something for us to think about, certainly from a microbial standpoint, is that when these things are under stress, they do like to spit stuff out, and cells will take stuff up. They're not evaluating what they're going to pick up, right? They take it up, and then it's a fitness question. But I think you raised an interesting point that underlies the stability question. I think it's something we as a field have not addressed enough, and that is that what we're putting in these cells really challenges these cells from a fitness standpoint. And I do think we have a lot of work to do from that standpoint, and to harness more so evolutionary dynamics in order to get these things to behave in the way we like. Okay, thank you, everyone.